Good morning to everyone. I am Dr.L.INBATHAMIZH. I am Assistant Professor, Department of Biotechnology, Satyabama Institute of Science and Technology, deemed to be University. I welcome you all to this session of lecture on basic concepts of enzymology. Now we'll come to the mechanism of action of enzymes. So when I say mechanism of action of enzyme, how do these enzymes act as biocatalysts? When I say catalyst, definitely these enzymes are involved in speeding up the rate of the reaction. So how are they going to increase the rate of reaction? For this, we have to think about the reaction in terms of its energetics. So when a reaction proceeds, the substrate or the reactants will get converted to products. So as they get converted to products, the substrate, when it moves to product, it will not occur in a horizontal line. It has to move up. So when it is moving up, it has to cross one hill of energy. Now the hill of energy or the excess energy which the substrate needs in order to get converted to products is called as activation energy. So only when the substrate can overcome or can increase its energy so as it can become products, we can expect the reaction to occur. But this may not happen in all cases, in which case we need to apply or make use of catalysts. So enzymes here, particularly in biological processes, when they are added, now they try to decrease the activation energy. So when they are decreasing the activation energy, the substrate need not have to climb up a hill. It can get converted to intermediate complexes called enzyme substrate complex. So this enzyme substrate complex will lower the activation energy hill and can easily convert the substrate into products. So this is how an enzyme catalyzed reaction can work, wherein it is trying to decrease the activation energy so that substrate can get easily converted to product, and thereby it is catalyzing or increasing the rate of the reaction. This is how a normal enzyme catalyzed reaction can work. Next, when we think about the properties of enzymes, as I told you earlier, enzymes are highly specific in nature and they depend upon certain important factors like pH, temperature and concentration. So when I talk about pH, as we all know, pH is negative logarithm of hydrogen ion concentration. So as you increase the pH, the rate of the reaction will increase. But after a particular point, after it reaches a peak, it falls down. So when it reaches a particular peak, the particular pH or the specific pH at which the enzyme activity is maximum, we call it as optimum pH. Now only at the optimum pH, the enzyme is fully active. On either sides of the slope, we expect the activity to be very, very less. So we can expect a bell-shaped curve for the effect of pH on enzyme activity. Similarly, if we move on to the effect of temperature, again we expect the same type of curve. The activity initially increases, then after reaching a particular point, it starts decreasing. Now this happens mainly because enzymes are proteins by nature. So obviously after you reach a particular point, the protein, that is the enzymes which are proteins, will get denatured. Thereby the activity decreases. Again here you can find a bell-shaped curve, and there is an optimum temperature, which is the temperature at which the enzyme activity is maximum. Here we need to know another term called as temperature coefficient. Now this is the rate of the reaction will double itself with every 10-degree rise in temperature. So this doubling of the rate with the increase in temperature we denote it by a term called temperature coefficient. Then moving on to the effect of substrate concentration on enzyme activity. Again, if you see, as you increase the concentration of the substrate, the rate of the enzyme activity increases. Again, it will start increasing, but after a particular point, it will remain stagnant, it remains constant. So the type of graph which you expect will be a rectangular hyperbola. Now why this rectangular hyperbola? Because after it reaches a particular point, the enzyme active site becomes saturated. It gets saturated with substrate molecules. So howsoever you keep adding the substrate, already the active site is completely filled. So the addition of substrate molecule is not going to affect the enzyme activity. Thereby the rate of the reaction remains constant. Here we need to know a term called Km value, which is called as Michaelis-Menten constant. Now this Km value is a substrate concentration when the rate of the reaction reaches half of the maximum velocity. Now this Km value is again a constant value for a particular enzyme. So this Km value gives a lot of information further in the enzyme studies. Now knowing about the factors like pH, temperature, substrate concentration, we should also know about the activity of the enzyme. So when we know about the activity of the enzyme, as there are molecules which can stimulate the activity, there are molecules which can as well decrease the activity of the enzyme. Thereby we need to know about the molecules called as inhibitors. So when I talk about inhibitors, again enzyme inhibitors, there are two main types. One is the reversible type, the other one is the irreversible type. The irreversible inhibitors will bind tightly to the active site so that their action cannot be reversed at any point of time. Now talking about the reversible inhibitors, these are flexible and the action can be reversed at any time. Now how we can classify these reversible inhibitors? There are three main categories. One is called as a competitive type, uncompetitive and non-competitive. Now how we can categorize these inhibitors? This is based upon where the inhibitor is going to bind to the enzyme. Is it going to be at the active site or at some other site? Or whether the enzyme action can be modulated or the inhibition can be reversed by increasing the concentration of substrate or not. If possible, where it is going to occur and how we are going to modulate, all that we can know under this particular reversible inhibitors. Now reversible, especially the competitive inhibitor, here the inhibitor resembles the structure of the substrate. Since it can resemble the structure of the substrate, at any point of time the inhibitor can compete with the substrate and it can bind to the active site. So there is always a competition between the inhibitor and the substrate to bind to the active site. For example, let me tell you about the action of succinate dehydrogenase. Here the substrate is succinate or succinic acid and the product is fumaric acid. Now there are few molecules which resemble succinic acid structurally. For example, malonic acid, oxalic acid, glutaric acid. If we see the structure of these molecules, they are more similar to the structure of succinic acid, which is the normal substrate for succinic acid dehydrogenase. So any point of time these inhibitor molecules will compete with succinic acid and will bind to the active site of succinic acid dehydrogenase. When they bind to the active site, obviously they will try to inhibit the action of the enzyme because we cannot expect the expected product. So this is what happens in competitive inhibition. And we have to remember here the inhibitor binds only to the active site. That is why there is a tough competition between the inhibitor and the substrate. Another example which is applicable, let me tell you, this is in case of a bacterial action, especially in the growth of bacteria. A particular enzyme folate synthetase is involved in the growth of bacteria. Here it requires a substrate called as para-aminobenzoic acid. Now this substrate is a normal substrate for the action of folate synthetase. But there is another molecule called as sulfanilic acid which resembles para-aminobenzoic acid structurally. So at any time sulfanilic acid, an amide form of it which is called a sulfanyl amide, can bind to the active site of the enzyme because the sulfanyl amide resembles para-aminobenzoic acid. Since it resembles para-aminobenzoic acid, it can bind to the active site of folate synthetase. When sulfanyl amide binds to the active site, it cannot allow para-aminobenzoic acid to bind to the active site. Obviously, the enzyme reaction is inhibited and folate is not synthesized. When folic acid is not synthesized, obviously it inhibits the growth of bacteria. This is how competitive inhibition works out. Then next about uncompetitive inhibition. Here the inhibitor is not going to bind to the active site. We can expect it to bind to the enzyme substrate complex because it is not going to bind directly to the enzyme. It is going to bind to the enzyme substrate complex. Similarly, there is another type called as non-competitive inhibitor where the inhibitor not only binds to the enzyme active site but also it can bind to enzyme substrate complex. So in these two cases, we cannot overcome the inhibition by increasing the concentration of substrate. Whereas in competitive inhibition, if we increase the concentration of substrate, definitely since it is a reversible type of inhibition, we can overcome the inhibition effect. Now let us move on to another type called as irreversible inhibition. Now the name itself suggests that the inhibition cannot be reversed, therefore it is highly rigid and the enzyme, once it is inhibited forever and we cannot retain back its activity. For example, let me tell you about iodoacetamide. Now this particular molecule acts as an irreversible inhibitor for the enzyme containing sulfhydryl groups. So when this molecule binds, it brings about a product which is going to totally inactivate the enzyme. Another example is diisopropyl fluorophosphate. Now this resembles sarin, which is a nerve gas used in World War, and this particular irreversible inhibitor binds to acetylcholine esterase, which is involved in hydrolyzing acetylcholine in neurotransmission. Since it is influencing neurotransmission, obviously it leads to the death of the person affected. So all these are the types of irreversible inhibitors. Another type of inhibition is feedback inhibition. This exists only in a particular case wherein the end product, when it is in excess, it will try to inhibit the initial step in the enzyme action. So since the end product is inhibiting the initial step, it is called as feedback inhibition. Thank you for the patient listening, and we'll meet back again with another interesting topic. Thank you.